if we use females with high genetic merit for fertility as donors, we do have an impact not only in the number of grade 1 embryos, but how those embryos perform once they are transferred.
Welcome back, everyone, to the Dairy Science Digest. This is a podcast designed to bring the Journal of Dairy Science straight to the ears of dairy producers. I'm Reagan Bluel from the University of Missouri Dairy Team, and today's guest is Dr. Sophia Ortega from the Department of Animal and Dairy Sciences at the University of Wisconsin. And today we're going to talk about pregnancy establishment, and specifically zooming in to the early embryonic stages and what the association of fertility traits are to that success. Now, our industry has really embraced these fertility traits such as cow and heifer conception rate and daughter pregnancy rate. So she and a team have conducted 2 retrospective trials to best understand how these performance parameters might impact the success of embryo transfer on a dairy operation. Ultimately, this will impact our bottom line when acknowledging the importance of just getting these gals pregnant in a timely fashion. These results were summarized in the featured article this month titled The Association of Fertility Traits with Embryonic Development and Pregnancy Establishment. So, Dr. Ortega, welcome to the Dairy Science Digest. Thank you so much for joining us today. Could you please take a moment to introduce yourself to the audience?
Hi, well, thank you, Reagan. Thank you so much for the invitation. I'm really excited to share some of my work. I'm right now an assistant professor at the Department of Animal and Dairy Sciences. I have a background in dairy, did my PhD at the University of Florida, actually working with fertility traits and embryo development. And I was a professor at the University of Missouri for 3 years, actually, and then moved to Wisconsin to keep working with dairy. So I'm very excited to share some of my work today. This work in particular is coming from a funded project by USDA. And we're trying to understand really, when we talk about fertility traits, what does that mean, right? We know, and it has been established already, that the animals with high genetic merit for fertility, they get pregnant faster. That has been established throughout. The question is, why? Because every genetic association, these fertility traits, right, or a pregnancy rate, which is a measure of days opens, and heifer conception rate or cow conception rate, which are measurements of the pregnancy to a specific service, right, to that service or that insemination. We know, right, high genetic values for these traits equals more pregnancies or faster pregnancies. And every genetic association, and these are genetic associations, these traits, right? But every one of these associations are relying on the phenotype that is measured. And this comes from data that is collected from the farm, from the Dairy Health Improvement Program, right? When dairies enroll, and they really fit into the CDCB database for trait calculations. I collaborate a lot with Dr. John Cole at the CDCB since I was a student, actually, and has been a fruitful collaboration so far. And one question is, can we improve these traits? Because we have a lot of information of these traits. Early pregnancy rate has been used for a couple of decades now, and and heifer conception rate and cow conception rate have been used probably more than 10 years now, around 10 years now. And the thing is that heritability of these traits is still low, around 4% and 1%. And the reason we cannot increase that reliability of these traits and heritability is because we don't have better phenotypes to do the associations, right? Against what phenotypes can we associate these traits? And this has been a burning question for me. How do we improve these traits? Because I don't think it's about just generating new traits up and down. These traits are already being calculated routinely. They are embedded in the net merit calculation. So when we select for high net merit, we are selecting for these traits as well. So how do we improve these traits to improve the overall selection for fertility? And when we think about pregnancy, one question is, why don't we break this down in pieces? How much is all the way to production of the embryo? How much is from the embryo to preg check?
Yes.
Right? That is the data that gets fed up in the system, right? Because what we get is the preg checks. And one of the biggest constraints that we have as researchers is access sometimes to data or large data sets. And I'm very excited about this study. It was an incredibly great collaboration with the URUS group, and they allow us to access to their large data sets and really plow through their data.
Tell me, who exactly is the URUS group, the URUS
Yeah,
group?
so the URUS group is a big holding, and right now under the URUS group, you have Big Genetics, who actually manage all the genetic improvement program for the holding, which also has Alta Genetics and Genex. So it's that entire group. So we had access to all the database. On the URUS group, you also find Transova Genetics. You will find BAS for all the dairy comp.
Yes.
So it's all this holding.
Okay, and so to kind of talk about that, guys, so she's taking this ginormous data set that actually is your data, right? Dairy farmers' data from both DRMS and dairy herd improvement, how that gets filtered in for the conception side of things, and then also URUS's giant data set completely anonymously, but then in a retrospective way looked at two different projects. Let's talk first a little bit about in vivo derived embryos versus in vitro procedures, because those are terms that you use often throughout your results. So can you describe what is the difference between IVD and IVP?
Yes, so the IVD or in vivo derived embryos, those are going to be animals that were subject to superstimulation, and then they were inseminated in the cow, and then the embryos flushed non-surgically 7 days later. So the entire early embryonic development happened inside the cow. In the case of IVP or IVF, IVP, those are in vitro produced embryos, and those are going to be females where the oocytes or the follicles were aspirated and the oocytes from the follicles transvaginally. That is by ovum pickup or OPU, which are terms you're going to see around literature. And then the oocytes were matured in vitro in the lab, fertilized in the lab, grown in the lab, and then we measured in the lab and they are transferred back into recipient cows.
Yeah, very good. And so I appreciate you giving us just a little bit of background there, because those of us that aren't always doing this, we're not really thinking in those terms. And so sometimes it's easy for me to get confused. Maybe the listeners are better off at that. And I want to draw attention to everybody to table 1, and that was looking at the difference between the IVP and IVD. And really, guys, grade 1 embryos are like the cat's meow. Those grade 1s tend to just be more successful. Really, the brass tacks of this is generating pregnancies that persist and create a calf. So can you tell us a little bit about the success of grade 1 embryos?
Yes, so this has been shown previously in smaller data sets. The grade 1 embryos, which are kind of the gold standard when you do evaluation of embryos. We use the evaluation following the guidelines from the International Embryo Technology Society. So a grade 1 embryo is that one that I would say is like Miss Universe, right, of the embryos. It's a beautiful embryo that is
We just love her.
formed, that doesn't show any particular defect. And what is interesting to us in this study, those embryos produced in vitro, so the oocyte collected from the cow, the embryo produced in the lab. In those grade 1 blastocysts, so the top tier of the embryos, and those transferred fresh into recipients, any commercial recipients, one thing that we found is that if those embryos came from high genetic merit for fertility donors, either for heifer conception rate or for daughter pregnancy rate, they do have higher pregnancy rates by day 30. And that is true when those embryos are transferred either to heifers or into cows. And this is very important because we are thinking about these are critical periods for embryo loss. Where after we have the formation of the blastocyst, from once it's transferred to the cow, that early placentation that occurs between day 20, 21, all the way to day 30, we have a good chunk of pregnancy loss there as well. That has been well documented. We try to monitor that with pregnancy-associated glycoproteins measurement of packs that we do sometimes in farms. And the fact that these embryos coming from high genetic merit for fertility females, they do better through this stage. So we have more animals that have remained pregnant by day 30, but that is also true.
And 60.
And 60.
Yeah. And it's not just more, guys. It's like 9% more, like a lot more. And they're all statistically significant with a p-value of really powerful. So we know definitively that you're going to see an improvement in the success of that pregnancy. Now, you did mention the cow and heifer side of things. Could you talk a little about the environment of the cow and how lactation might impact the success of this?
Yes, so we know, right, in particular in high-producing dairy cows, they are subject to a lot of more of lactation stress, right, particularly in early lactation period. So they are dealing with a lot more sometimes than some heifers, and that's why we wanted to tease out if that effect was true in cows and in heifers, right? Because we might see it in heifers just because those animals are not under lactation physiology stress, but we also see it in cows. So that means that these embryos are resilient in both environments, and I think that's a very important tale. But there are studies, right, where first lactation heifers actually do have great fertility. That's work that has been done here at Wisconsin and at Cornell as well. But yeah, when we think about the cow, they are in a completely different nutritional plane, production plane. Yeah. They have to deal with a lot more than a heifer. And I think having this effect show in both cows and heifers shows that is specific embryo effect, right, and genetics effect.
Yes. I'm so excited to see all of these animals hit the ground and become mature cows, and think about the actual progress that we as a dairy herd, the United States dairy herd, might be making in reproductive success, right? We're just a few years away from really seeing this explode.
Yes, actually right now the USA is the highest in vitro embryo producer in the world. For a long time, this was Brazil's territory. And from the last few years, that the US has really taken the lead.
Okay, so this is going to go beyond the parameters of our country then. This is going to go global, isn't it?
Yes, and think about it. These traits are used globally. Like we use it here in the US, but even though in other countries they will have other fertility traits, the base is sometimes the same, right? And there are a lot of comparison between these traits with other use in other countries. But a lot of the US exports genetics to other places, right? So a lot of our sires, semen, estrus, goes to several countries. So the impact of our genetic improvement does have impact in other places as well.
Yeah, so humbling.
And we have that tendency that if we select for higher genetic merit for fertility values for the donors, we are going to have more grade 1 embryos when we do in vivo embryo production. And that has to do, I think, with that maternal environment, really.
Sure, yes. Her oocytes are translating that. So specifically drawing numbers to what your statement is, you were observing a 13.6% increase in grade 1 embryos for those that are in vivo. That's incredible. That's a big number. And I love that you say Miss Universe. Like, yes, we love these grade 1 embryos because of how successful they tend to be. That's a gold standard for embryo technology. Can we pause for just a minute and talk about where and how embryo transfer has started to infiltrate the dairy industry more here in the last, say, 5 to 10 years? Can you kind of give us a background about the improvement or change and increase in embryo transfers?
Yes, sure. So we definitely are seeing a change in the trend. If we see the statistics from the International Embryo Technology Society and the American Embryo Transfer Association, there is a shift in terms of the use of embryo technologies in general in dairies, right? And actually there is a shift from the in vivo produced embryos, so those that are produced inside the cow, to the in vitro produced embryos, those that are produced inside the lab, in the lab. And part of that is handling, right? And cost and results and homogenizations of procedures. The in vitro produced embryos are really taking up. I think more than 1 million embryos are produced in the US. And I'm pretty sure that's the statistics from last year from the International Embryo Technology Society, and that now has surpassed, right, that in vivo embryo production.
Wow.
But if you think about the logistics, right, if you have to do all these inseminations in the cow after superovulation, and it's not bad, it works all good, but requires an extra logistic. And now we have more access. There are many commercial labs around the country that can help with the in vitro embryo production.
Sure.
There are some advantages on, for example, the use of sires. You can use the same straw to produce hundreds of embryos, and that presents some opportunities to invest in higher quality sires or specific sires to certain productions, or even pulling certain oocytes. So there are some advantages to in vitro produce embryos. One thing that has been an issue is actually producing grade 1 embryos. Sometimes we have a fewer yield of grade 1 embryos in vitro than we have in vivo. And again, it's just because the in vitro system, we have improved it a lot, but it's still an artificial system in the lab, right?
And that's where Dr. Ortega says, here I come. I'm going to come help answer that question.
And,
Yes.
and the thing is, we need to acknowledge the limitations of each model and each technique, right? So even though that in vitro embryo production has improved greatly, we don't still match the number of blastocysts or sometimes the quality of blastocysts that are produced in vivo. However, I think the system has improved quite a bit in the last few years. And when we produce grade 1 embryos and we transfer, we're seeing 50% pregnancy rates in some cases, and those are very normal now to see for in vitro produced embryos. But the question I had is, so how do we better select the females that are going to produce these in vitro, these grade 1 embryos? And that's kind of how this study came about.
Okay. And so really zooming in then on these fertility traits, focusing around, your heifer conception rate and daughter pregnancy rate. How did these traits shine when you were looking at blastocyst rate? Or I guess first my question is, what is blastocyst rate and how did these fertility traits impact it?
Yes. So a blastocyst rate is when you put a number of oocytes that are fertilized in vitro, and then we put them through culture. After 7 days, they will form a structure that is called a blastocyst. In a blastocyst, we'll have different layers of cells now. We have a defined layer of cells that will form the placenta, and we have a defined structure that will form the embryo proper. And that's what we want, that normal development, right? That happens both inside the cow, and now we can do it in the lab. Usually we cannot grow embryos beyond that stage. They have to go back to the cow. We cannot do that really in vitro beyond the blastocyst stage. So what we found is the number of blastocysts, and I will check actually the number of blastocysts produced in vivo when you have higher values on the females or the donors for heifer conception rate, we have more blastocysts in vivo, which means that out of the follicles that were counted when animals were super stimulated and inseminated, we ended up having more blastocysts recovered in the flush. That's what that meant. When we did it in vitro, we did not find an increase in the number of blastocysts. So from all the oocytes that were collected and they were put into culture, we didn't find an increase in the number of blastocysts. So that percentage was very consistent. And that pretty much is talking about the difference in the maternal environment, right? When those embryos are growing inside the cow, we just have more that develop to the blastocyst stage.
And so that is directly correlated to that fertility of mama, and so that the fertility of the donor is enhancing the success of that blastocyst. Is that correct?
Yes.
Yeah, and so there's a lot of talk about on the donor side, and I know that's a big feature here, but what about the recipients? I know that wasn't necessarily covered here in the paper, but just in your workings, because I know this is kind of your wheelhouse, can you talk about problem breeders receiving embryos and what that might look like?
Yes, so in this case, when we usually use embryo technologies, we try to have healthy recipients, but we don't worry as much about the genetics of the recipient, right? Because that's exactly one of the points of using embryo technologies, that you can use your higher genetic females as donors to produce the embryos, and then use the rest of the hairs or other recipients that are not genetically so valuable for the farm as recipients. And in this case, we did have a small group of animals. I think there were like 200, a little bit above 200 recipients for which we had genetic merit. And we didn't find an association for the genetic merit of the recipient and pregnancy. Mm-hmm. But in that case, it was a very small group.
Yeah.
That is difficult because most of the embryos are transferred in commercial farms with sometimes non-genotype recipients.
I see.
So there are opportunities, right? The use of embryo technologies have different opportunities, like I said, to use recipients of lesser genetic value, and also it can be used in crossbreds, right? You can have your high production of high genetic merit embryos and transfer into some crossbreds that you might have, and really still move forward with pure genetics or the genetics that you want in any setting.
While retaining that cow that is so valuable as a uterus right now. You know, she's just a home for that desirable embryo. Good.
Exactly. Awesome. And it can be like even changing, right? In this case, we're thinking about specific breeds, but it can be even for switching your hair from one breed to another. Embryo technologies allow you to do that really quickly.
Absolutely. So you're really zooming in on the daughter pregnancy rate, heifer conception rate, and cow conception rate. Are those found in net merit?
Yes. So these traits are calculated from all the data that come from the DHI program, the MRS, right, into the CDCB. That's what we get in the evaluations that are released by the CDCB multiple times a year, but they are also embedded in net merit. So when we select for higher net merit in a female or in a male, we are selecting for fertility as well. That's why index selection is always recommended, right? They're selected for a specific trait. The thing with these traits is, I think I mentioned before, they have lower heritability. So meaning that what we know right now, little is controlled by the genes. I think that has not been able to improve as much just because we don't have better phenotypes to associate with. And that's what we're trying to do with this type of studies, breaking down pregnancy and trying to find those specific phenotypes. And what we're going to do next is now trying to go to the genotypes. We needed to find first, do we have a phenotype at the embryo level associated with these traits? Now that we know that we do, we're going to hopefully move forward trying to evaluate what are the specific genetic variants associated with these phenotypes and traits so we can improve the heritability and reliability of these traits.
So cool. Very cool. And so it's obviously very clear why USDA would want to support your program, ma'am, because ultimately the work that you're doing is being documented, again, through the CDCB, which is the Council on Dairy Cattle Breeding. And I'll go ahead and put a link in the show notes for that. And it's a great conglomerate of information about the genetic progress of our dairy industry. And it's hard work that you're doing and your team's doing that's made these incredible advancements to the dairy industry. I mean, guys, if you just pause and think about how far the dairy industry has come, even since the 90s, but that line is so steeply increasing because of the work of all the geneticists out there and reproductive specialists, and you can't deny the nutrition side of it too. So, I mean, it's just incredible what this beast is doing, and I can't wait to see what the next 5 years will unlock.
I'm excited. Like we actually put a proposal for the following up of this now going to the genetics. So we are hoping that we get lucky with the USDA,
sure,
because now it's trying to find what are the genes, right? These are the genes that control this specific process. So then we can use these genes to calculate the traits. So then that's the way that we're going to improve heritability and reliability.
Yes, you bet. So does that look like, so the grant will then pay the commercial producers to take genetic analysis then? Is that the way you're going with it?
So the way we go through that is also through the CDCB, because those animals are already genotyped, true. But we need to have access to that and funding to be able to do all the analysis, and that's what we're trying to get to. What is beautiful about this proposal is really potentiating the use of the information that is already generated.
Yes.
It's not about just, that's what I have a lot of friends, right? They do, let's do another trait. It's not about doing new traits. It's really understanding the ones we have, improving the ones we have, because they are well established.
Right.
how the information is collected, how they get calculated is well established. Can we improve that?
Sure.
So improving our system will have a more direct impact than trying to establish a new trait, right? And starting reinventing something. Right. So what this will allow, if we are lucky to get funded, is really trying to grab all that information, now the genetics. But that's the thing. We needed to know, do we have a phenotype first? Right. Because if we don't have it, it makes no sense to go there.
It wouldn't. Yeah.
But now we have it.
You've got it. Yeah. Those are some really strong p-values. You've got it, right?
Yes.
That's exciting.
And a lot of transfers, right? We have like 5,000 transfers for the other one. Actually, I think the calculations for the embryo production is like 10,000 procedures. So that really allows us to have confidence in how the data that we're presenting.
And I love that because it's almost like citizen science, right? This is the dairy industry, pouring information into your hands so that you can then unlock that information and give it back.
Be useful.
And make it useful. So thank you for the science that you do. And thank you, the Land Grant University, for being able to facilitate this knowledge that then directly impacts the dairy farmer, And
I think one thing that is important to recognize here is that we really, at this case, had the support of the industry, right? We work as islands, so we are the researcher here at the university, and then is the industry here, and they can do progress privately, right? Mm-hmm. But having that access and having that merge, it really makes an impact for their program, but also for all the producers, because at the end, all these industries work with farmers across the U.S., right? And that's the genetics that is getting poured into the farmers as well.
That's good.
And I think that's a beautiful marriage, and it's difficult for that to happen, but we were very lucky that they were very helpful.
That's awesome. It's perfect. Yeah, and so just to reiterate that the enrollment in DRMS, or Dairy Records Management Service,
and the DHIA, the Dairy Health Improvement
Association, I'll go ahead and put clickable links to those also in the show notes. And so if you want to be part of this citizen science, please just enroll your herd. You'll be amazed with the data that you gather up from those pieces of information. So, Dr. Ortega, if there was one particular thing out of this very informative project that you really want boots on the ground dairy producers to really understand, what would that be?
First of all, of course, keep working with the MRS, DHI, right? Because when you enroll in these programs and feed into all these large data sets, they really help us trying to understand these traits and try to improve them. And will go back to you, right? As we improve these traits and you use all the indexes and traits provided by the CDCB for your own selection in your herds, all this comes together. You get results from all the work and how you participate in this program. And that's really important, because us alone as researchers cannot do it, right? We need help from all these programs to get good science out and trying to understand this. So also knowing that there's always been a little taboo, right? That if we have a low heritability trait, we do not improve. And I think one thing that this is showing is that you can still improve, even when the heritability of these traits is low compared to milk production and other traits. You can still select for fertility. You can still improve reproduction using genetic selection. We're trying to get the impact of that improvement higher and the magnitude higher, but you can still select for it. Definitely using these traits to evaluate your donors is a tool now available, right? Like if you have 100 females that have high genetic merit or good genetic merit, and you want to do some filter in terms of which are going to be my embryo donors, if you decide to use embryo technologies, you can look at these traits as well, right?
For sure.
As an extra kick, because now we know that if we use females with high genetic merit for fertility as donors, we do have an impact not only in the number of grade 1 embryos, but how those embryos perform once they are transferred. And I think at the end of the day, we all want more calves on the ground.
You bet.
And that's why we went, and we were very lucky to have access to this database to follow all these pregnancies to having calvings, right? And be able to evaluate the calf rate. It was very intriguing to us and very important to see that if you grab an embryo, and we did that analysis, they have high genetic merit for fertility, like a parental average from what is coming from the mom and what is coming from the dad. And you transfer that embryo to any recipient, any commercial recipient, you still have more calves on the ground with a high genetic merit for fertility embryo than one that is with low.
Yeah, that's what matters.
And I think that's what you want to know, right? Which are the ones that I want to transfer, or from which females. That's why we presented both the females and the parental average of the embryo. But in both cases, if you use a high genetic merit for fertility donor for embryo technologies, you are going to have likely more calves on the ground.
Beautiful. Beautiful. Well, Sophia, this has been a great discussion, and I want to thank you so much for your time and willingness to share this deep dive into embryo transfer and the development of those little embryos and what might help switch success on and off. I know this will help dairy producers better understand how to potentially select future females for flushing. Listeners, I applaud you for taking time out of your day today to listen in on this conversation and better understand these early stages of reproductive success. I've really enjoyed our conversation and this discussion. I've learned so much. This has been the March edition of the Dairy Science Digest, which is a monthly podcast project designed to bring the Journal of Dairy Science straight to the ears of dairy producers. We highlight peer-reviewed research articles actively in press because it's sound science that you can base your management decisions around. And of course, it's provided to you by your University of Missouri Dairy team. So please be sure to like, share, and subscribe to get future editions straight to your cell phone. This is Reagan Bluel with the Dairy Science Digest, and I hope you have a great day.